In the 1990s, plant breeders began working with a new technology to develop breeding lines in a shorter time frame. This was known as doubled haploid plant production. Since then, this has become a valuable tool to plant breeders across the world. But what exactly is a doubled haploid? The term haploid refers to a single set of chromosomes. A plant's gametes are haploid cells, and when they unite in fertilization, a diploid embryo is formed. If instead the chromosomes in one of those haploid cells can be doubled, a doubled haploid organism is produced, where both chromosomes of a pair carry identical alleles at all the genes. To better understand this new technology, let's turn to some experts from Colorado State University's Wheat Breeding and Genetics program. First, we'll hear a little bit about traditional wheat breeding methods before diving into the doubled haploid approach.
Traditional breeding starts with cross-pollination between two parents with complementary traits. For example, a high-yielding but disease-susceptible parent crossed to one with good disease resistance. Each parent contributes one set of chromosomes to produce an F1 plant. For the genes that differ between the parents, the F1 is 0% homozygous, meaning the alleles differ between sets of chromosomes. The F1 is then self-pollinated to create the F2 generation. During meiosis, genetic recombination occurs. Because the crossover points between chromosomes occur randomly, this results in a range of F2 plants that contain different combinations of parental chromosome segments. One or more of these plants is selected for further breeding through self-pollination. Each generation is more homozygous than the last, so by F6, the expected homozygosity is 96.9%. Depending on the breeding method, it may take 3 to 6 years for wheat to arrive at the nearly uniform F6 stage.
The doubled haploid method was developed to reduce the time needed to achieve homozygosity. With this technology, breeders can obtain uniform breeding lines in just half the time. While there are a variety of different approaches, the one we'll explore is used at the Wheat Breeding and Genetics program to produce new lines of winter wheat.
Similar to traditional breeding, the doubled haploid method starts by crossing two wheat parents with complementary traits to create an F1 plant. Our approach is based on wide hybridization, which is the process of crossing between distantly related plants. For this method, we have to prevent our F1 wheat from self-pollinating and instead pollinate it with corn.
When head is ready for emasculation, so emasculation means removal of all anthers. We keep only like female organs, and we pollinate those female reproductive system. We are fertilized by corn pollen. So like when we do emasculation, after 1 or 2 days, the stigma, it becomes receptive. So I take the pollen from corn, I collect the corn pollen on a dish, then I was like shake over the emasculated head. And after 1 day or within 24 to 48 hours, we put some hormone there, 2,4-D, well, to make the embryo bigger.
The corn chromosomes are eliminated after successive cell divisions, leaving only a single haploid set of the female wheat chromosomes that combine segments from the original two wheat parents. Since the embryo would otherwise abort, embryo rescue is used to coax it into surviving. Embryo rescue starts 2 to 3 weeks after pollination.
So then we cut them and take to the laboratory, put in water, and we take the seed out. Then we sterilize the seeds and do embryo culture under the sterile hood. These are hybrid seeds, and she will take out under the dissection microscope that embryo. One by one, she cuts it, take the embryo out, and put in media. These are still haploid, and this is ready to go to vernalization. Because it is a winter wheat, we have to put in vernalization for 2 months. After 2 months, I take out.
After vernalization, exposure to temperatures of 4 degrees Celsius or less, these seedlings are then transferred to the greenhouse and grown for about 2 weeks. And it's the next step that's key to doubling the chromosomes in these haploid plants.
So we do colchicine treatment here. Those plants, I take out from the soil. I wash them and cut to almost skipping 1 inch root. and put the colchicine there, so the root will be like dipped in the colchicine. I do overnight, and like haploid number chromosome become double.
Colchicine is a mitotic inhibitor. By inhibiting chromosome segregation during cell division, daughter cells are expected to have double the number of chromosomes.
After doubling the chromosomes, the next step is to plant the wheat in the greenhouse for seed production. A few days after planting, the tips of the leaves start to burn and turn brown. We trim that dead material off, then grow the plants in high humidity for several weeks. The first generation of doubled haploids often produces heads that are not fully filled with seed. Since seed-producing heads come from tillers, it's important to promote tiller growth using fertilizer and by keeping the soil loose between plants. It takes about 4 months for the plants to become ripe and ready to harvest.
After we produce the doubled haploids, we bring them out to the field. So generally we get about 10 to 20 seed for each doubled haploid. Take that out to the field and we plant it as a small 2-row plot. And this 2-row plot allows us to produce enough seed to go into replicated yield testing the next year. And so in this 2-row plot, we're looking for a number of different things. So just general agronomic traits. So how tall is it? When does it flower? When does it mature? Is it producing, good spikes? Is it producing a lot of tillers? If there's diseases present, we try to select for resistance to those diseases or resistance to pests as well. Generally, we get population sizes of around 100 lines. In general, we would probably keep maybe 20% of those lines to advance into yield testing. The ones that make the cut, that look good, we would come in and we would harvest this row, and that allows us enough seed to plant in 6 or 7 locations the next year. The doubled haploids allow us to speed up inbred line development. We produce enough seed to go straight into replicated yield testing, which is a big advantage for the program.
Now that we've seen the process of creating doubled haploid wheat, let's find out how this technology can benefit wheat breeding programs.
I think what probably most wheat breeders or most plant breeders would think about would be the reduction in cycle time that we can achieve through the use of doubled haploid technology. And what I mean by that is the time that it takes us to make a cross between two different wheat varieties, derive a new line from that cross, and then use that line in another cross for a next cycle of breeding is what we refer to as cycle time. And so we can essentially cut that in half. There's other advantages. I guess the first one that comes to mind is the simplification of seed purification, which we all have to do in applied plant breeding programs. And then also, I guess, thinking about it from the producer's perspective is being able to respond faster to producer constraints. And one example that I would have for that would be the development of Fortify SF, which is a wheat stem sawfly tolerant variety that we were able to develop at CSU in just 6 years' time.
In addition to speeding up the breeding process, creating doubled haploids may offer additional benefits for those researching plant genetics.
If we think about plant breeding, our objective is to make genetic gain. And so we have used doubled haploids to bring in traits from synthetic wheats and develop some doubled haploids from that. And we may not get a variety out of that, but we're crossing it to an adapted parent. We get a new parent that's more adapted, that has that new source of genetic variation, and we're able to have that parent and use it within a shorter time frame. So I think they're a valuable tool for bringing in new sources of genetic variation. Another thing we do with doubled haploids coming out of the greenhouse is we genotype them and then use that sequencing information to be able to predict traits that we don't see out in the field. So if we don't have stripe rust, well, we have the ability now to predict how a line is going to perform against stripe rust, so how resistant it's going to be. We can also predict grain yield using a method called genomic selection. So if you have an interesting trait in your breeding material, you can develop a mapping population to study that trait and try to identify the genome position of loci that control that trait. And this has been done on a lot of cool traits in the program. Wheat streak mosaic virus would be a good example. Wheat curl mite resistance, and more recently, resistance to wheat stem sawfly.
While doubled haploid technology has been a boon to CSU's wheat breeding and genetics program, there are some drawbacks to relying on this technique, and some reasons why it might not be the right choice for every program.
Because of the cost and because of all the resource constraints in developing double haploids, we essentially have to restrict the crosses that we would use for making double haploids. And so we pick our best crosses, what we think are our best crosses. And in that process, we reduce the amount of, genetic diversity that we may have in the breeding program.
Potential downside is just the amount of recombination you get compared to developing an inbred line over 5 years. And so with doubled haploids, you're essentially fixing that line in the F2 stage. And so you're only getting really one generation of effective recombination.
Despite these limitations, doubled haploid technology can be extremely beneficial as long as you can provide the right laboratory, greenhouse, and field conditions for your crops.
A regular procedure like fertilizer application, pesticide application, and watering, they should not experience dry condition. An excellent greenhouse condition, which is like disease and insect free, because that condition will give you only healthy plants. And if plants are not healthy, we do not get good seed, no good embryo, not good regeneration. So everything, all steps will be affected.
Above all else, a skilled and dedicated team of scientists is what is most needed to make a double haploid program successful.
People talk about plant breeding as being an art and a science, but being successful at developing doubled haploids is every bit the art that we have just in plant breeding in general. And it's having good people that are good with the plants, people that are good with tissue culture. And Dr. Santra and Hong Wang are really the best that I would say in terms of making that all work. And not just their capacity, but their dedication. That has been the thing that I think has been most important for us in terms of success of the effort.
So that's double haploids for you. A process that can create uniform breeding lines in half the time of traditional methods. It allows plant breeders to be more adaptable to the needs of the time, since they can more quickly produce lines with greater yield or lines that address the ever-evolving problems caused by pests, disease, and a changing climate.